Hello students, welcome to Bachelor of Science program, third year, subject Botany, semester 5, course code BOD105, course title Nursery and Gardening. The title of this unit is Seed and the module name is Seed Banks. I am Miss Aditi Venkatesh Naik, Assistant Professor from PES College of Arts and Science, Farmagudi. The outline of this course comprises of introduction to seed banks and the role of seed banks in conservation of biodiversity. The learning outcomes, students will be able to state the importance of seed banks and they'll be also able to describe the role of seed banks in conservation of biodiversity. Now, what is a seed bank? A seed bank is a repository that stores germplasm as seeds in order to maintain the genetic variety or genotypes for the future. Now, the term seed bank sometimes refers also to a cryogenic laboratory facility in which the seeds of certain species can be preserved for up to a century or more without losing their fertility or viability. The seed banks are often flood, explosion, and radiation proof valves that house jars of seeds from various plant species. Here, the seeds are normally stored at low humidity and cold temperatures, about minus 20 degrees Celsius. Now, because of these conditions, it helps the seeds, it protects the seeds, ensuring that they grow when needed later on in future. Now, these seed banks can also be referred to as a special type of arboretum or like gardens where seeds are harvested and the crop is rotated. The plants that cannot be preserved in seed banks. The only option for preserving such germplasm of plants is the in vitro storage, where cuttings of the plants are kept under strict conditions like in glass tubes or vessels with the use of biotechnological tools. There are over 1,000 seed banks that exist around the world, which differ in their type, the size, and the purpose. For example, the Millennium Seed Bank in Sussex, which is managed and directed by the Royal Botanical Gardens Kew, is the world's largest seed bank available. This seed bank, that is the Millennium Seed Bank, was launched in the year 2000 and has seeds from approximately 40,000 different species from all over the world, including nearly all the UK's native trees and plants. Now, this slide shows the images of the Millennium Seed Bank at Wakehurst Place, West Sussex, in England. These are the buildings which exist and these are the conditions in which the jars containing the seeds are kept at strict environmental conditions which favor the preservation of the seeds which can be later used for germination under special conditions. This image shows the International Crop Research Institute for the Semi-Arid Tropics Seed Bank, which hosts around 1,25,000 accessions or seeds which are collected and assembled from over 140 countries. And this seed bank serves as the world's repository for collection of germplasm from 6 mandate crops like sorghum, millets, peas, 5 different types of small millets like foxtail, little millet, kodo millet, and so on. It manages the gene banks which, in India, Niger, Kenya, and Zimbabwe. When we see the seed banks, in order to maintain uniformity over the world, follows techniques which are well developed for crop plants by organizations such as the International Plant Genetic Resources Institute, that is IPGRI, which was previously known as the International Board of Plant Genetic Resources, that is IBPGR, and the Food and Agricultural Organization of the United Nations. The main advantage of seed banking is that it allows the large populations of plant species to be preserved and preventing or minimizing the genetic erosion by providing optimum conditions and reducing the need for regeneration. We all know regeneration takes a very long time, and hence there is the seed banks have advantage over regeneration, where large populations can be preserved and genetic erosion can be minimized. The seed banks are the most cost effective method which provides plant genetic resources for long term ex situ conservation, that is, conservation that occurs outside the natural habitat. The seed conservation is quite easy, relatively safe, and needs minimum space, unlike that of conserving plants in botanical gardens. So, seed conservation or conservation of plants through the seeds is quite easy, relatively safe, and needs minimal space. Let us see seed bank as conservation of genetic diversity. Natural restoration of plants and preservation of endangered plants may be preserved through seed banks or germplasm banks. However, if natural population still exist, it may be preferable to recollect rather than regenerate a fresh supply from the previous collection, because damage such as mutations associated with the viability or loss due to storage can occur. We see that if natural populations are still existing, we need not go into storing the seeds and using the same for propagation. Rather, it is preferred to recollect or recollecting the seeds from a fresh supply rather than using the previous collection, because damage may occur or mutations may also occur which are associated with viability of the seeds. Now, the success of the long term seed conservation is dependent on continual viability monitoring and regeneration or recollection when the viability of the sample forms below a certain threshold. It means that it is important to understand or to monitor the seeds that have been kept in the seed banks, because after a period of time the seeds may lose their viability, and hence the seeds need to be monitored continuously and have to be regenerated. And if there is loss of viability or the amount of regeneration from the seeds is less, then one needs to go for recollection, that is recollecting or going and collecting again when the viability of the sample falls below a certain threshold which has been laid down by standards. It is critical to recognize that no matter how carefully the seed collection has taken place, the regeneration and storage was performed, natural selection cannot be imitated or mimicked, and sometimes artificial selection is unavoidable, which results in unforeseen genetic changes. Thus, the major consideration for long term conservation of germplasm collections is the determination of the seed behavior of each individual species to be preserved during storage under dry and cold temperatures. So, each species or each seed may show differential behavior to the kind of preservation that has been carried out, or the type of conditions that a seed has been kept in order to utilize it for long term storage. Hence, it is important that each of the seed behavior has to be determined for each of the seeds. Now, thus, we see that there are various activities that have been carried out by the seed banks. Firstly, the collection of the seeds. The seeds are prepared, that is, seed preparation. The seeds are dried or drying of seeds. The seeds are packaged and then they are and restorage, which is followed by periodical germination tests in order to see and understand the viability of the seeds, and then plant seed regeneration, recollection and restorage and documentation. Documentation is a very important aspect of seed banks. When we look at the seeds are categorized into two different types, that is orthodox seeds and recalcitrant seeds. Orthodox seeds are the seeds that can be dried to a low moisture content and stored at low temperatures without losing its viability for long periods of time. Example: seeds of corn, wheat, rice, carrot, papaya, pepper, chickpea, cotton, sunflower. Now, seeds of these orthodox seeds, like the examples that I've just said, if we dry these seeds, we all know that such seeds, which are orthodox seeds, they can be dried in order to and dehydrated, and then after a long period of time they can be germinated. They are, and we see that the seeds are still viable, whereas the recalcitrant seeds are seeds which show very drastic loss in viability, which with decrease in moisture content below 12 to 13 percentage. Example: citrus, cocoa, coffee, rubber, oil palm, mango, jackfruit, and so on. Let us now understand the storage of recalcitrant seeds. The plant, a species of recalcitrant or long-term conservation, cannot be achieved through seed banking because different methods have been developed with respective merits. As I've already said, that recalcitrant seeds show very drastic loss in viability with decrease in moisture content, and because of this, they cannot be stored with the help of seed banks. And for such plants or such seeds, we need to develop or look into different methods with respect to their respective merits. Like, one can look at alternatively for field gene banks, in vitro germplasm collections, pollen and DNA banks. In many cases, it is the main problem which is being faced by botanical gardens dealing with different plant species, of which a great proportion of the plants or seeds are recalcitrant. Therefore, we need to develop, or there is a need to develop, complementary conservation techniques and adopt different methods for the recalcitrant seeds. Whereas storage of orthodox seeds is possible with the aid of seed banks. Your long-term seed storage of orthodox species, as recommended by IPGRI, is to dry the seeds to a moisture content of below 7% and seal the dry seeds in a moisture-proof container such as laminated foil bags, aluminum cans, or glass jars for storage at a low temperature of minus 18 degrees Celsius. Clearly, this is only applied to true orthodox species of plants and their relatives. However, because less known about wild species, a temperature of about minus 4 degrees Celsius and moisture content of 7 to 8% is advisable to begin with. In case of orthodox seeds, if the seeds are dried to a low percentage humidity, such as they have been dried below 8%, the seeds will be able to withstand very cold temperatures, that is below 20 degrees Celsius in majority of the cases. This has been tried and tested, and it is seen that if the seeds are dried to a very low percentage or very low percent of humidity, such as below 8%, the seeds are able to withstand very cold temperatures. Therefore, the orthodox seeds are able to tolerate extreme cold conditions and can still remain viable after many years of storage, and hence are desiccation tolerant as opposed to the recalcitrant seeds which are intolerant to desiccation. However, that there exist some orthodox seeds that cannot be maintained in seed banks, such as they show very low seed production or long life cycles, such as trees and perennials, where their conservation as seeds would make the study of their biology difficult. Hence, we see that the seeds which show very long life cycles, such as trees and perennials, their conservation as seeds in the seed banks might be difficult because during the periodic germination test and the plant regeneration test, which is a continuous process in the seed banks, such seeds which are being obtained from trees which are perennials or which show long life cycles is quite difficult. Let us look at examples of plants which are plants or seeds rather which are stored through the seed banks. One example is Indian clover, that is Trifolium amoenum, which was thought to be extinct initially, but in the year 1993 was rediscovered by Peter Connors in the form of a single plant which was existing in western Sonoma country. Peter Connors harvested these seeds and he grew the specimens of this critically endangered species in a controlled environment. The second example is Wollemi pine, which is another example of the plant that is being preserved via ex situ conservation, as they are being grown in nurseries and are sold to general public with the help of seed banks. Let us conclude the storing seeds and the ex situ way or associating with the supportive seed banks in countries around the globe. Every seed bank, if we see, gives a safe home to some of the world's most threatened plants. Now, with the help and support of the seed banks, seeds can be germinated and reintroduced back into the wild and can be used for scientific research in finding our future food and medicines. All thanks to seed banks. Let us summarize. The seed bank is nothing but a place where seeds are stored to preserve genetic diversity for future. These seed banks are usually flood, bomb, radiation-proof valves which holds jars of seeds from different plant species. It is an effective method of providing plant genetic resources for long-term ex situ conservation, especially in case of endangered plants, which allows preservation of populations for up to a century or more without losing their viability or fertility. However, it is of utmost importance that one determines the seed behavior of each individual species, which is a significant factor. The ex situ conservation of orthodox seeds is achieved by drying seeds to a low moisture content and storing these seeds at very low temperatures, which helps in keeping them, storing them for a very long periods of time without losing their viability. Whereas the ex situ conservation of recalcitrant seeds is not possible or it's difficult via the seed banks, and this can be achieved by field gene banks, in vitro storage methods, pollen banks, and DNA banks. These are some of the references that students can refer to. Thank you so much.